Alright everyone, so today we're going to do chapter 3, aerobic gram-positive cocci. In the book you guys will see mainly clinical cases, and then the boxes pretty much has just a nice overview of like the facts that you guys need to know about these organisms. So here we go. Objectives, same as usual. Describe the general characteristics of Staphylococcus, Streptococcus, Enterococcus, including oxygenation, microscopic gram staining, characteristics, and macroscopic appearance on blood agar. Recall the genus and species of the most important pathogens in this group. List the media used for the isolation and differentiation of Staphylococci, Streptococci, and Enterococci. Identify the key biochemical reactions to identify the clinically significant Staphylococci, Streptococci, and Enterococci, and then explain the chemical principle associated with each test. And then define MRSA as it relates to antibiotic susceptibility and the methods used to control transmission of the organisms. And then list various types of diseases specifically associated with Staphylococcus, Streptococcus, and Enterococcus. List the virulence factors. Explain the significance of group B strep in perinatal infections. Describe the colonial morphologic characteristics, clinical significance, and laboratory techniques for the identification and recovery of nutritionally variant streptococci. And then identify a clinical isolate based on the results from standard laboratory diagnostic procedures. I know it's a lot of words, but trust me, we will be able to go over all of this stuff in just this little lecture. All right, so general characteristics of your Staphylococcus aureus, which is the main organism that we're kind of concerned about when it comes to our Staphylococci. These are gram-positive cocci, so purple. Catalase positive, which is a biochemical test. They are non-modal. They are aerobic or facultatively anaerobic. And then on blood agar, they're usually medium-sized, circular, smooth, convex, and are white to yellow in colony. So here's just a little picture of what they should look like under the gram stain. A couple things you should know. Epidemiology and pathogenesis is that they colonize by either entering, adhering, or establishing infection. Basically, they already live on you, so it's really easy for them to cause an infection because they're already right there on you. They can multiply by avoiding host immune responses, interfering with the host immune response, or damaging tissues via toxins. Staphylococci and Streptococci both have toxins that can cause some serious issues. Transmission can also be person to person. And the other thing about these guys is that they can also produce a slime layer or a biofilm. And then, of course, that peptidoglycan layer, which makes them purple, and the protein A in the cell wall also contributes to their pathogenesis. There are actually a lot of toxins, but at this point you guys don't need to know them. You just need to know that there are toxins. Spectrum of disease, basically they can cause localized skin infections. There are 4 different ones listed here, and you guys should know the difference between those 4 as well. Toxin-mediated disease. So these are some of the ones that can be caused due to the toxins and not just the staph alone. Scalded skin syndrome and toxic shock syndrome. And then, of course, systemic disease can be caused by Staph aureus as well. Some specific organisms that you guys should know. In your book, it does not list Micrococcus. However, just kind of start bringing that into your mind because we will be covering that. So those are the ones that are usually, in fact, those who are immunocompromised. Then you've got Staph aureus, again with this soft and skin and soft tissue infections. Your toxic shock syndrome, food poisoning, and scalded skin syndrome. Staph sarcophagus is usually associated with UTIs, urinary tract infections, in sexually active young women. So that's a little factoid that you should remember. And then S. epidermidis is usually tied to nosocomial infections. Media, we normally will use sheep blood agar, which is the routine medium for this organism. There's something called mannitol salt agar, which you guys will actually be making in lab. And it just has a high concentration of the sugar mannitol and salt. And then a phenol red dye, which helps us determine, which is a color indicator for pH. There's another type of medium called PEA, CNA, and chrome agar. Chrome agar is usually used for the identification of MRSA. The two biochemical tests that you guys should know for staphylococci and streptococci is your catalase and coagulase test. The catalase test is the one that distinguishes staph from strep, and then the coagulase test distinguishes Staph aureus from other staphylococci. The coagulase test is actually an enzyme that clots plasma, or it uses an enzyme that clots plasma. And so again, Staph aureus is positive, and then everything else is pretty much negative. So the ones that you guys should know are your Staph epidermidis and your Staph saprophagus. And these are also commonly called coagulase negative staph, or coag negative staph for short. MRSA, methicillin-resistant Staphylococcus aureus, is basically just what it is. It's resistant to methicillin. And this strain of Staph aureus carries the gene MECA, or the MECA gene. Methicillin resistance can also occur in other staph, and then infections are usually then treated with vancomycin. Prevention. Remember I was telling you guys before that staph already lives in you, and so it's really, really important that we, observe proper hand hygiene. For those who are intranasal carriers of S. aureus are usually treated with antibiotics. Newborns are then treated with antibiotics to prevent the spread in the nursery. And then the CDC also recommends that they screen patients for MRSA before being admitted to the healthcare setting. Again, because it's resistant to methicillin, so and it likes to stick around, and it's quite hardy. So it's really important that we try to keep control it. On to our streptococci. General characteristics here are they are also gram-positive. They can appear in chains, so like you see in the picture here, little chains or long chains, and also individually or in pairs. They are facultatively anaerobic. They have varying hemolytic patterns, alpha, beta, and gamma. We will get into that in just a minute. They are non-modal, and the colonies are usually small to medium, circular, and range from transparent to off-white or gray. Your most common pathogens in your streptococci and enterococci are your strep pyogenes, so group A, strep agalactiae, or group B, strep pneumoniae, and then enterococcus faecium and faecalis, and also some virulent streptococci. So here are the different hemolysis patterns that you can see. This is kind of like a nice picture. So alpha hemolysis is partial hemolysis. And if you look at the picture, this one right here, you can kind of see that there's a greening of the media. So this is blood agar, first of all. And what it does is it turns the blood agar into this greenish color in the background, so to say. And that's what we call alpha hemolysis. Beta hemolysis is complete lysis. Okay, so right here. And you see how you can kind of see through the agar. If you were to look at this and pretend like you were holding this up to the light, you would be able to see the light shining through. And again, that's complete lysis of the RBCs in the blood agar. And then the last one down here is your gamma hemolysis, and as you can tell, there's no hemolysis of the red cells in the agar at all. So again, these are normally part of our normal human flora. So they are normally found in your pharynx, mouth, lower GI tract, or vagina, but can cause life-threatening infection when introduced into sterile sites. So blood, CSF, other body fluids, bone, vascular tissue. Clinical infections. Strep pyogenes will cause a strep throat, actually, or bacterial pharyngitis. It also causes skin infections, scarlet fever, and something called post-streptococcal disease. And basically what happens is if strep pyogenes, or if you have strep throat and it doesn't get treated, you can actually get a lot sicker later on as it travels through your system. Then we have strep agalactiae. For you guys, what's most important to know is that it can cause meningitis in newborns. It's normally not considered pathogenic unless we recover it from a pregnant female or female of childbearing age, sometimes even. And then we have strep pneumoniae, which causes pneumonia. And then your Enterococcus species, which can cause also urinary tract infections, bacteremia, and intra-abdominal infections. Virulence factors. You really just have to be able to list these at this point. When we get into next semester, you guys will have to do a little bit more explaining when it comes to these guys. But for now, just start getting familiar with what the virulence factors are. Then later on, we'll get into more in-depth information. This kind of just goes into it a little bit. Don't really worry about the slide too much, but just if you were interested, again, what you need to know is that Group B is associated, or Group B infections are associated with neonates. Early onset, where symptoms occur within the first 5 days of life, and you can also have late onset, where symptoms occur a week to 3 months after birth. Strep pneumoniae is most infections with pneumococcus cause pneumonia and meningitis with or without bacteremia. These also these appear as gram-positive diplococci and gram stain. And the two main virulence factors that you should be aware of with these two is that it has a polysaccharide capsule, which inhibits phagocytosis, and then a pneumolysin, which can affect the host cells. Enterococcus is typically not as virulent as your other gram-positive cocci, but here is just, again, a list of those. And again, you don't really have to know too much about it, just be able to list some. Prevention and control for your streptococci. There is only actually one vaccine, and that is for pneumonia. That's called that pneumococcal vaccine. I don't know if you guys have kind of heard on the news where, people don't get vaccinated and, or the babies don't get vaccinated, and so when they get infected, they can die like pretty fast, or can get sick really, really fast as well. And then, of course, prevention is to make sure that you isolate somebody who has pneumonia. And that is it for this chapter.